Welcome to Lakshman Biology. Now we will discuss about monohybrid cross or monohybridization. Monohybridization. This monohybridization also called as inheritance of one gene. Inheritance. Inheritance means transfer of characters from generation to generation. Inheritance of one gene also called as monohybrid cross. Inheritance of one gene. Once again, if you see the same thing, Mendel. Mendel is the scientist. He did work on the Pisum sativum or garden pea plant. If you see the Mendel work, why he did the work? To know about how characters are transferred from one generation to another generation. Actually, he proposed the three laws to explain how characters are transferred from one generation to another generation. First two laws, law of dominance, law of segregation. Both he identified. First law, second law, that will explain about how characters are transferred from one generation to another generation. He explained by using monohybrid cross or monohybridization, or it is also called as inheritance of one gene. Inheritance of one gene. First of all, if you see the definition of monohybrid cross or monohybridization means cross between pure lines. Pure lines means which contain homozygous condition. Those are formed by continuous selfing. It may contain either homozygous dominant or they may contain homozygous recessive. Those are called as pure lines. Those are not in the heterozygous condition. Cross between pure lines which differ means differentiate. Cross between pure lines which differ in single character. Single character. That is called as monohybrid cross. Cross between pure lines which differ in single character is called as monohybrid cross. In the monohybrid cross, what Mendel did, he have taken the one character, that is a height of plant. Okay, single character. What is a single character? He taken height of the plant. If you observe the height of the plant, height of the plant is character. Already we know that the Mendel selected the contrasting traits. The contrasting traits of this character is, one is the tall, another one is dwarf. Tall, another one is dwarf. If you see the tall plant and dwarf plant, these are pure lines because which contain only homozygous condition. The genotype of tall plant is capital T, capital T. The genotype of dwarf plant is small t, small t. Capital T, homozygous dominant. Small t, homozygous recessive. Homozygous dominant tall plant crossed with the homozygous recessive dwarf plant. Then, see, during the formation of gametes, actually, when we cross the two plants, first what we can see, we are going to cross the gametes. We are going to mix the gametes. If you observe the gametes, how gametes are formed. During formation of gametes, only one allele. There are two alleles in the homozygous dominant. There are two alleles in the homozygous recessive. In these two alleles, only one allele is enter into each gamete. Okay, because reduction division we can observe during the formation of gametes. Hence, few gametes contain capital T, another 50% gametes also contain capital T. These alleles are separated during formation of gametes. Then, if you see the recessive plant, homozygous recessive plant, which is also produce the gametes which contain only one allele, that is small t. Then, when you see the genotype of F1 generation, this is the gametes. After the fusion of gametes, you can see the formation of new generation, that is called as F1 generation. This F1 generation we can also call as filial generation, first filial generation. If you observe the first filial generation, if you see first filial generation here, if see here, this is the T, this is the T, this is the T, capital T, small t. Then if you observe the genotype from this capital T, small t, capital T, small t, capital T, small t, all contain heterozygous, heterozygous, heterozygous dominant genotype. Already we know that the dominant allele is the allele which can express its phenotypic character even in the presence of another alternative allele. Hence, all dominant alleles, each genotype plant contain one dominant allele. Hence, you can see all tall plants. Here, all plants are tall. The F1 generation contain all plants, all tall plants. But the difference here, genotype, heterozygous genotype we can observe in the F1 generation. If you see the tall plant which present in the parent generation, parental generation, these are the parents, we can see homozygous dominant condition. When you cross the F1 generation, that is only called selfing. Selfing. Selfing is nothing but crossing of the plants which are obtained from the same parents. Okay, same generation and that should be obtained from the same parents. See, when you cross the F1 generation, we can get the F2 generation. After crossing F1 generation, we can get the F2 generation. After crossing the parents plants, we can get the F1 generation. After crossing the F1 generation, we can get the F2 generation. If you observe the genotype, first gametes which are formed in plants, which are gametes which are produced by the F1 generation plants is, here you can see the two types of alleles. Hence, 50% alleles contain dominant, 50% gametes contain dominant allele, under 50% gametes contain recessive allele. Here also same, two types of gametes are formed. 50% contain dominant allele, another 50% contain recessive allele. Then if you see the, when the gamete, this gamete will mix with this gamete, we can see the formation of homozygous dominant. When these two gametes are mixed, we can see formation of heterozygous dominant. Here also you can see heterozygous dominant, capital T, small t. Here small t, both are mixed. Homozygous recessive. If you observe here, we can see the formation of three tall plants. 1, 2, 3. Three tall plants. Number of tall plants are 3. Next, if you see the number of dwarf plants, TT, 1 only. One dwarf plant, one tall plant. This is the phenotypic ratio. Phenotype. Phenotype is nothing but the appearance, morphological appearance. What we can see from externally, that is the phenotype. Three tall plants, one dwarf plant. Then if you see the genotype, genotypic ratio, genotype, that is the genetic constitution. We can see 1 is to 2 is to 1 ratio. Genotype is 1 is to 2 is to 1 ratio. 1 is capital T, homozygous dominant. 2 heterozygous dominant we can observe. 1 homozygous recessive we can see. This is the genotypic ratio. Once again, if you observe the phenotypic ratio, 3 is to 1. If you observe the genotypic ratio, that is 1 is to 2 is to 1. That is about the monohybrid cross or monohybridization. See, actually, Mendel did work with many plants. Mendel did work with many plants by taking the different types of characters. One time he taken the seed shape and crossed. One time he have taken the pod shape and crossed. One time he taken seed color and crossed. Every time he got the result, that is the phenotypic ratio 3 is to 1, genotypic ratio is 1 is to 2 is to 1. In next class, we will see what he identified after the getting result monohybrid cross. That we have to see. Thank you.